Okay, welcome back. Let's talk about the immune system today. The system that helps us not get sick, which is very important. We are under constant attack. We need to defend against bacteria, viruses, fungi, parasites, cancer toxins, and other pollutants. The best line of defense, guess what? Is to not let the stuff come into the system. So we have skin, we have mucous membranes that protect us by forming a boundary between the inside and the outside environment. Remember, a boundary is a need to be able to survive and live. Once an invader is inside the system, so they've breached that, then we have two internal mechanisms to fend off invaders. We have a non-specific immunity that attacks the invaders non-selectively. It just generally shoots them down. That's mediated or triggered by the inflammatory response. And then we got specific immunity, and that's when we have pathogens that we have previously seen, previously encountered, the system has to act on them already and is sensitized to them. Like, for example, we have chickenpox and we get exposed to it, so we get sick, but then the system learns that pathology. And after second exposure, it can react to it way faster, and we don't really get sick. And the system works sort of subliminally on its own. It goes very fast. So that's the specific immunity that's learned. That requires, though, that we have a first exposure to learn what the pathogen looks like. Both of those systems, the selective and the specific and the non-specific, have cells, and we describe them in the cellular immunity as well as soluble factors, and we'll talk about them when we talk about in terms of humoral immunity. And we use both of those to combat the trespassers. In terms of the non-specific immunity, the trigger is the inflammatory response. So that's what triggers the activation of the factors that come in and help fix the problem. When we look at the top picture there in the top right, when we have a tissue injury, we have physical agents come in like dirt or so, chemical agents as well as pathogens. In terms of the system, what it does, it vasodilates the capillaries that are in the area of trouble. So we have a capillary widening, so we have an increased blood flow to that area. Then it increases the capillary permeability. That means the capillaries in that area become leaky. And sometimes they're actually damaged, so it just leaks out anyway. So that's a release of more fluid in the area. Those two are great because that walls off the problem and it doesn't go further into the system. Then the chemicals, in terms of the inflammatory chemicals in that area, will attract white blood cells to that area. So they migrate there. That's a process that's done via chemotaxis. We talked previously about that in the blood system, or the blood chapter. We have neutrophils, monocytes, which then on the side of action become phagocytic macrophages. And we also actually have lymphocytes that will travel to that area. A process called chemotaxis. The neutrophils are the ones that first go to action and they phagocytize, they eat the material, and then they render that harmless, of course, because it's inside a cell, it's not floating around anymore. The neutrophils themselves, they will perish and they will produce pus. So that's when you see pus, you know it's that neutrophils, you know they kill themselves for you, you know you can be grateful for that. If you feel depressed, you know they love you. But we got a few other things in the non-selective, non-specific immunity that I want to touch on. The first things are natural killer cells. They're NK cells. And they are specialized lymphocytes that attack pathogens and tumor cells by perforating the membrane with specific proteins called perforins. So what they actually do is they go in and they shoot down the target. They shoot out proteins that look like bullets, and they are perforins. T cells work very similarly. T cells in our system are the sharpshooters that sort of target one specific pathogen invader and shoot that one down. That's the T cells. We'll talk about them. They are very, very dangerous. They're very important, but they need to be kept in check all the time. So otherwise, they'll attack our own cells, and we don't like that. Macrophages are interesting. Macrophages are sort of the link between the non-specific and the specific immunity. So what they do is they eat big stuff. Macro is big, right? So they ingest a pathogen that helps prevent further damage because it's inside a cell now. But then what the macrophages does, it combines the identifying bits of bad stuff, the antigen, that will be the antigen. We talked about that in the bloods. The antigen is the thing that you see, or that is a shape that is recognizable as an individual bad thing, or an individual thing. Here is just a bad thing. And then it combines that with a protein of its own self, of its own, and that's the major histocompatibility complex. We'll talk about that next. And then when they present the MHC, major histocompatibility complex, and the antigen combined, they present that to the T lymphocytes, which are the sharpshooters, and they will then shoot down or learn who to shoot down. Very interesting stuff. Macrophages also produce cytokines, and these are signal proteins. Kines are kind of localized hormonal action chemicals, and so they act locally. So they secrete those cytokines to activate and coordinate cells of the non-specific immune system. For example, the cytokines are the things that will create a fever, and those are endogenous pyrogens. And fever is here because some of the bacteria don't like higher temperatures, so we can actually mitigate their replication that way. Also, what is standing by is interesting. We have a system of plasma proteins, and they can aggregate in a complement system and perforate bacterial cell walls through what they call a membrane attack complex. So that's similar to the perforance. It's just a whole hole is created in a cell membrane, and once the boundaries breach between the inside and the outside, that cell will perish. The major histocompatibility complex are individually distinct cell surface proteins that allow the immune system to recognize when a microorganism hides in the body cell. The MHC combined with fragments of our own self protein signal to the rest of the crowd, leave us alone, it's our own cell. But if that MHC is combined with an antigen, as we see with the antigen-presenting cell in the previous slide, the infected cell betrays the invaders by sounding the molecular alarm. It says, hey, we got it. I've recognized it. I've been invaded. And then the immune system can attack because that's a molecular alarm. It's very, very important, though, to understand who is self and who is not self. So the immune system learns at birth the difference between self and foreign material. Everything it touches at birth is tolerated and recognized as self, and everything that is coming after birth is recognized as foreign and needs to be attacked. The problem with that is, for example, when we have organ transplants, that's foreign, so we need to attack it. And then what that means, we reject it. Failure of this self-non-self recognition or distinction results in an immune system that is an autoimmune system. So we actually have antibodies, which are part of that humoral immunity. Those are protein-based, and they fail to understand the self-non-self differential distinction, and then that results in an autoimmune disease in which the antibodies are formed against the own proteins and attack them. And that, of course, is a problem. All right, let's do that as the first section. We'll go to the second section in a minute.